We're going to talk a little bit here about biochemistry. This is about as technical as we're going to get in nutrition. What is biochemistry? It is the study of life at a molecular level. There are three basic areas or major areas of biochemistry that we look at when we're talking about nutrition. We look at structure, which is the three-dimensional arrangement of the atoms within that molecule. We look at metabolism. How is it produced and used for energy in our body? We also look at bioinformation, which is how this is transferred from RNA to DNA. And in that, we're going to talk about biomolecules, which are just any molecule that is produced by a living cell or an organism. So some of the fundamentals are the basics that we've talked about, you've talked about. We have atoms in a compound that are held together by, there's two different types of possible chemical bonds. We could have an ionic bond, which is a bond that's between a metal ion and a negatively charged non-metal ion. Hydroxyapatite in enamel is composed of ionic bonds between calcium, phosphate, and hydroxide ions. So that's an example of an ionic bond. Covalent bonds are when the electrons are shared between two non-metals. So those are the two different types of bonds that we'll discuss. We have functional groups. So functional groups are groups of atoms that will give a specific molecule its characteristics. It makes it what it is. It can be converted into other functional groups through chemical reactions. So by oxidation, which is a loss of an electron, or by reduction, which is where a molecule will gain an electron, or an atom will. Biomolecules can be degraded or synthesized through either one of these reactions. Condensation is another reaction that can occur, and this is where a new molecule is formed between two smaller molecules. So I think of condensation on a window. It's forming a water molecule from hydrogen and oxygen. Hydrolysis or hydrolysis, I always think hydrolysis. Lysis means to cut or to split. So if you've got a hydro, that's like a water molecule, we're going to break that into molecules, into two smaller molecules. So that would kind of be the opposite of condensation. It's the breakdown of that water molecule. Here are some common functional groups that make a molecule the molecule that it is. If it's an alcohol, it's got an oxygen and a hydrogen. Amine groups or amino would be proteins, and that would have a nitrogen component. So these are all just the little functional groups that attach themselves to the molecule that make that molecule what it is. There are four basic major classes of biomolecules. We've got carbohydrates, proteins, nucleic acids, and lipids. Carbohydrates are going to be the ones we discuss first. They are 4 kilocalories per gram. They can be classified as monosaccharides, or they can be disaccharides or polysaccharides. So we've got simple carbs and complex carbohydrates. The simple carbohydrates are going to fall under the monosaccharide or disaccharide category. Those are going to be the carbohydrates that we probably all enjoy the most, but they're not quite as good for us. So cookies and candies and cakes and sugars like that. Polysaccharides are many. It's a sugar chain composed of many. Di would be 2 and mono would be 1. So polysaccharides are going to be our complex carbohydrates, like your starches, your potatoes, your grains, things like that, that tend to be a little bit better choice for us when we're selecting carbohydrates. And their biological function, carbohydrates' biological functions are, for number 1, energy metabolism. Carbohydrates are broken down into glucose and many other sugars too, but glucose is the source of energy for the body. That's the preferred source of energy for our body, especially your brain and central nervous system. So that is very important. Carbohydrates can also become a form of energy and be stored. So these are some of the common monosaccharides that you might see. Glucose, again, that's the sugar that our body prefers. Galactose is a milk derivative. Fructose is the sweetest of the monosaccharides, and it comes from fruit. And then sorbose is another monosaccharide. Disaccharides would be the combination of two. So a glucose and a glucose make maltose. A galactose and a glucose make lactose. And glucose and fructose make sucrose, which is what you would know as table sugar. Protein has its own purpose in our body. Proteins are they're the same. They're 4 kilocalories per gram. So carbohydrates and proteins both provide the same amount of energy or calories per gram, and that is 4. They are essential for almost every physiological function in our body. They help provide structure. They are important for muscle contraction. They help transport and store substances. Reactions occur because of proteins. They help regulate our metabolism and support our immune system, and they are composed of building blocks called amino acids. Carbohydrates are many times known to be protein sparing because they help our bodies conserve protein rather than using the protein for energy. Our body prefers to use carbohydrates, and that way our body can reserve that protein to repair and grow and do all the things that it needs to do in our body. Muscle contraction, provide structure, reactions, so that we don't utilize our protein inefficiently. Enzymes are helpful in helping proteins catalyze these reactions. So proteases begin the process of digestion of proteins. So you can kind of see that if you look at your prefix there, it's pro. So that's for proteins. The only one that's a little harder to reason is the amylase. But we talk about salivary amylase is the first breakdown of carbohydrates in our body. As we take in carbohydrates, the salivary amylase begins that process, and then the amylase enzymes in the body continue the process of digesting dietary carbohydrates. Lipases begin the process of breaking down dietary fats, and we know that lipo or lipids means fat, so the others are kind of easy to guess what their job might be. They must, in order for an enzyme to catalyze or help catalyze reactions, they have to bind to a specific substrate. I always think of this as like a puzzle piece fitting into the right spot. There's a specific site called the active site on an enzyme, and it has to bind or connect with that specific substrate in order to catalyze that reaction. It's almost like a key that once that key gets put in place, it starts the engine. Coenzymes are assist enzymes with reactions. So kind of like a co-worker. They are vitamins. Many of the B vitamins are coenzymes to the use and digestion of carbohydrates and proteins. They're non-protein organic substances, and they are regenerated at the end of a lot of these reactions. What are vitamins? Vitamins are essential nutrients that we need in our diet because we can't synthesize vitamins. They are often referred to as vital amines. Many water-soluble vitamins are precursors to those coenzymes I talked about, so B vitamins and vitamin C. And then the fat-soluble vitamins are A, D, E, and K. Fat-soluble vitamins can accumulate in our fat stores and cell membranes, and they are not involved as coenzymes. So DNA and RNA store and transfer our genetic information. We know this. What is a genome? It is the genetic information needed for the development of an organism. There's been lots of scientific study on genomes. I've heard a lot of the scientists that are working on the vaccination for COVID referring to genome studies. They're learning lots from this. Why do certain genetic makeups react to these viruses the way that they do, or to drugs the way they do? So that's very important. Nucleic acids are composed of building blocks called nucleotides. The general structure of a nucleotide is a sugar. It can be ribose if it's RNA, or deoxyribose if it's DNA. And these are attached to nitrogenous bases and a phosphate. And that's just what they look like. That double helix is DNA, where there's two twisted, and then RNA is a single helix. And they're, the thiamine and uracil are the different nitrogenous base in RNA versus DNA. RNA is really important in protein synthesis. So it carries messages from the DNA to the ribosome. It transfers that and carries that amino acid to the ribosome to incorporate that into the growing polypeptide chain. So anytime I see RNA, ribose, I think protein. Fats. Fats are also called lipids, and they are higher calories, 9 kilocalories per gram in fat. Fats involved in energy metabolism and storage. It provides structural components of our membranes, our biological membranes. It's also very important for insulation and protection. It acts as hormones that help regulate our body. It serves as vitamins because we have 4 fat-soluble vitamins, A, D, E, and K. And it acts as a detergent in digestion. So it kind of helps in getting our nutrients through for digestion. There are fatty acids, which are the structural components in complex lipids. They can be classified as saturated, unsaturated, monounsaturated, or polyunsaturated. So the presence of saturated fats, they contain single carbon bonds. And the more hydrogens that are attached to the carbon bond, the more saturated an element is. Unsaturated fatty acids are healthier fats. The mono obviously is contain only one carbon bond, and then polyunsaturated contain more than one carbon bond. And those are your fish oils, nuts, avocado, olives, things like that. Those fats that we consider to be healthier for our bodies. Triglycerides are the metabolic fuel in our body for our cells. It is the storage form of fatty acids. It is stored in fat tissue. Triglycerides are, they can be a solid at room temperature, or they can be a liquid at room temperature. If they're a solid, they can be saturated. And if they're liquid, they're predominantly unsaturated. If you think of coconut oil, that is, it's a solid at room temperature. I think people get kind of confused sometimes as to whether those that are solid at room temperature that aren't an animal fat, if those are still healthy fats or not. Hydrogenation, you've probably heard about trans fats and hydrogenated fats and things like that, because we're learning more and more that at one point we thought this was a healthier option. They would take vegetable oils that were a liquid at room temperature, and by adding hydrogen molecules to those, it converts basically an unsaturated fat to a saturated fat. But it was vegetable oil, so we thought it was healthy. We find out that that's not exactly the healthiest choice for us. So partially hydrogenated vegetable oil can be used to create that semi-soft fat product, which was really nice because we wanted it not to be liquid and we want it not to be as hard as butter out of the refrigerator. So this was like a good combination of both. It was spreadable and people liked working with that or cooking with that. But then we find out that it resulted in trans fats. So that is not a good thing. We want to watch our trans fats and hydrogenated products and try to keep that to a minimum along with saturated. Some antioxidants have been added to vegetable oils to prevent rancidity. So vitamin C and vitamin E sometimes are added to those to keep them fresh so that they don't become spoiled. Hydrogenation, this is kind of just showing you how the hydrogens are added. If you look at the first example, there's 2 hydrogens, and in the next example there's 4 hydrogens, and then they are transposed in the lower where they have transposed where those hydrogen atoms are located, and that would be a trans fat. Steroids. This is another class of lipids. Cholesterol is the most abundant steroid in our body, and it is a precursor to hormones such as progesterone, estradiol, testosterone, bile acids, and soluble vitamin D. Vitamin D can also be called a hormone. There are lipoproteins in our body that help transport the cholesterol through our blood. We've got high-density lipoproteins and low-density lipoproteins. High-density lipoproteins are desirable. The more high-density lipoproteins we have, the better. They are higher in protein and lower in fat, where low-density lipoproteins, I always remember, are lousy, and they are higher in fat and lower in protein. How cells can acquire, transform, store, and use energy is pretty much the definition of metabolism. So it's the sum total of all the chemical reactions in our body. We've got coordination of these reactions, we've got regulation, we have energy requirements for that to occur. As nutrients are oxidized in catabolism or breakdown, electrons are released from the metabolic pathways through glycolysis and the Krebs cycle. Remember, Krebs cycle gives off carbon dioxide, ATP, and water. The process of catabolism, or the breakdown, is energy producing. So this is the breakdown of complex molecules into smaller molecules, and it also involves the oxidation of those metabolites. Anabolism is building, so we require energy to build. This is the process of the biosynthesis of complex biomolecules from simple molecules, and in this particular case we've got the opposite. We have the reduction of metabolites. So here's kind of a flow chart of how the whole thing begins. We have our energy source, which is ATP, and we need ATP in order to build an anabolic reaction. Then we will see muscle contraction, transport, biosynthesis of cellular components, and then we will use energy to degrade that or break down carbohydrates, proteins, and lipids, which is a catabolic reaction, which is energy producing, and then we go right back in our loop again and start over with our energy source, which is ATP, which we need glucose in our body to provide. So carbohydrate metabolism is a complete aerobic oxidation of glucose through the Krebs cycle, producing carbon dioxide, water, and ATP. Excessive amounts of glucose can be stored in our muscles and our liver as glycogen, and that can be called upon if need be. There are 3 hormones that help regulate carbohydrate metabolism. Insulin, as we know, is very important for combating an influx of carbohydrates. So this would be present in a fed state. Glucagon is what is present in a starved state. So if we are low on carbohydrates, glucagon will appear. And then epinephrine is another hormone that we might see, and this would be when our bodies are in that fight or flight situation where we need immediate energy, where the body shuts down all of the unnecessary processes and focuses all of its energy on, if you need to run, on your muscles, on your leg, on your heart, on those kinds of things. So that would be epinephrine. And here is a flow chart with your carbohydrates. Like I said, they're simple. Those break down into either mono or disaccharides and then down into the sugars that fall under those categories. Whereas complex carbohydrates, which are the healthier choice, are your polysaccharides, which are your starches, your fibers, and glycogen, which is the stored form of glucose, falls under complex carbohydrate. So protein metabolism provides amino acid for the production of new proteins in our body. Amino acids are a source of nitrogen, which is helpful for the production of hormones, heme in our blood, as well as the nitrogenous bases that make up our DNA and RNA. There are ketogenic amino acids that will break down into acetyl-CoA that are then converted into ketone bodies. They can be converted into glucose for fuel for the body, which carbohydrates are the preferred source. Our body can't make all 20 amino acids, so some of them we have to get from our diet. These in your book are also referred to as indispensable. So indispensable amino acids are essential in the diet. The non-essential amino acids are the body makes those. They're also called dispensable amino acids. So these are the ones that we don't have to get from our diet. Our body will produce these on its own. Like I said, fats or lipids are a major source of energy because they have 9 kilocalories per gram versus carbohydrates and protein at only 4 kilocalories per gram. So it is, like I said, a major source of energy for the body. It is excess dietary fats, though, as we know, are stored in our adipose or fat cells as triglycerides. So when we're fasting or starving or dealing with an untreated diabetic or low-carb diets, there can be an excessive degradation of fatty acids, which can produce too much acetyl-CoA or those ketone bodies. And you guys have heard of all these keto diets where people are going high protein, low carbohydrates. So what they're doing by putting their bodies into ketosis is burning fat for energy rather than carbohydrates. Nutrigenomics is a scientific study of how foods and their components will interact with our genes. So eventually, the hope is that we'll be able to characterize diet-related chronic diseases and predict the responses to changing our nutrition. Hopefully, we'll have more control over some of these by making those changes. Genomics is the mapping or sequencing and analyzing of the genome that we talked about earlier, which is our entire DNA sequence that makes up an organism. So they're focusing on prevention and control of these chronic diseases by looking at our genome and then applying that to nutrition and how can we combat some of these diseases in the future. Some of the influences over our health. Sometimes we can't always help when we have these issues. Diabetic people, you can see somebody who appears to be healthy, they exercise, they eat right, but yet they're still diabetic. That could have been a genetic issue. It can be very powerful. It can be passed on from generation to generation. A person's nutrition, obviously. Emotions. Stress is a huge factor in health and can affect how our bodies respond to different diseases and conditions. Age, our lifestyle, and environmental factors all influence a person's health. What is epigenetics? Is the study of the changes in gene expression. So this can be active versus inactive genes. It does not involve changes to the underlying DNA. So an example of this might be the epigenetic changes that affect folate-dependent enzymes that increase the risk for developmental conditions such as neural tube defects. So that's an example of an epigenetic change. So in this nutrigenomic that we were talking about, where nutrition can affect how our genes react to these chronic diseases, we hope that someday it will provide the basis for being able to personalize dietary recommendations for individuals and their genetic makeup. And maybe we could even get to the point where chronic diseases could be preventable or delayed. And those that are identified as being at risk for some of these diseases might be more motivated to follow that specific dietary regimen if they know for sure that it can reverse, stop, delay the onset of some of these diseases.